Okay. I used to be great at my exams.
Did that help the scores for students or made it worse? Did that generally improve the average or depress the average? Great. Independently. Okay. That's good.
I'm in a good mood. Yeah. I think you probably get a little more. Then I make fun of you in front of no one understands.
But then they're not chemical engineers, so they understand even less than we do. Yeah. So then we look smart, right? Smarter.
Let's see, at the same time, jokes.
Something over because we think we know.
Okay.
So they don't think they know, so they might get it.
How many molecules of water per second go through aquaporin? Now don't look in your notes. Look in your notes. I already saw it. Okay. How about you? You don't even have those. Molecules per second. Water molecules per second going through these little things here. Okay. Yeah, so at any rate, our guest speaker, as typical in your jobs when you actually get a real job where they're paying you, don't always work out as smoothly. It was working really good up until yesterday. He may even have to miss the Ohio State games now under the installation of Bioproduction, which he's not happy about. But he makes at least twice my salary. So, especially when he goes to Canada, jumps out of helicopters to snowboard down mountains. Yeah, so my sympathy for him doesn't exist. So Aquaporin, back to you since you're you don't get off the 10,000. Okay, well you got a 10 part of it, so that's like a 5 in a good mood. But it's like 10 here. 10 to 10. Okay, so yeah, a little off. Water molecules per second. I was just trying to remember, again, I spent a lot of time back when they let me teach 340, and we'll continue next year. Trying to actually make biology like physically meaningful. Again, the thing that I mentioned before I did this was told you, what's the diffusional limit for like glucose hydrolyzed molecules per second for one enzyme number. Number the second would be. No, I'm just saying it's 100. Just pointing out, just in case you don't know. Okay. So that's it's a tiny number. Again, it's the number these people throw around 40,000 molecules per second. Okay, and you can actually get to that from a calculation of diffusion. How fast is it? I'll give you an idea. Now, diffusion's slow. It seems really slow, but it's not slow when you're talking about just moving a couple dozen angstroms, right? And that's what you're talking about. Getting it in there, have Pac-Man chew on it, spit it back out. Okay, so 40,000 molecules per second is a lot. Okay, and it starts to give you an idea of what it takes to actually make bio work. Bio does stuff it has to do it on a molecular basis, but it's minuscule compared to, say, methane on a platinum catalyst or something there, because it's a high temperature gas-phase reaction. Diffusion limited, what do you do? Right, with the temperature, right? The path length goes up, so we get away with that as chemical engineers. It's not fast enough, just heat it up. Okay? Biological systems, heat it up, we're dead. Okay? Doesn't work. So again, just reiterating a lot of themes throughout this thing that'll be the basis of what I hope you actually eventually get. And I threw out this, this particular one. It is where we lost. I'm not going to spend too much time. There's a couple of things. The idea behind this, and I'd love to go back and assign a homework time, but I won't. Ask. You can find, for instance, the water requirements of a city. Pick your favorite city. New York. You like New York? Better not Pennsylvania City. That's just terrible. Okay, yeah, okay. Especially New York. That's horrifying. Why is New York actually physically? I told you about the whole pumping and all the junk that was required for Philadelphia. How is New York City? There's some truth there. They had the water there in the first place. There's an amazing, you've never seen the giant tunneling machines that actually made these giant things. They through 20-foot diameter. And they went the whole way up into upstate New York, and there's giant reservoirs up there that have these. That's what gets the water into New York City. That's why New York City can exist with such a density, is its ability to basically tap into. I forget. It's amazing documentaries of these boring machines. They can do the giant 20-foot things through solid granite, and that's what brings the water into the city, and why it can actually exist. If you take a look then at something like, then you could translate that to how many aquaporin pores would be necessary if you could make a biomimetic membrane, where you could put that onto there, use a block of polymer or something like that to give you the laminates. How much protein would you have to make to supply the water for a good-sized city? I won't go to New York, it's a little extreme. Well, actually, it's not so extreme. I think I did it once for LA. Right? Yes. What are we talking about? What are units of mass? Give me a unit. Grams. Good. Okay. Do you think it's a gram? Probably more than a gram. Okay. Then let's just go on up. What are the big numbers? Kilograms. Okay. Or how about tons? Stuff like that. It turns out, no, it's actually in the grams. I don't even know if it's in the kilograms. So if you had that much protein, it would be enough to purify the water usage requirements. That's how fast water is going to do that. And trying to put that into an actual context. And so it gives you something to think about. Other assignments I used to do would be like, translating thermal heat loads for a city to how much water would evaporate relative to your favorite lake in the world. Okay. And so how much of that lake is necessary? It actually gives you some physical feeling for what's going on in the world on a scale that's enormous. And yet, that's still pretty dramatic. We're talking about a kilogram of protein to purify all the water for a city. I don't know what LA is. I'm going to guess it's less than 10 million, since that's closer to the New York City size. There's not much. Small issue. So going on further, I won't go into the whole. This is all molecular biology crap, okay, which you guys all learned in 340 ad nauseum. Maybe not. Not my problem. But the basic idea is you got to clone it into there. I mentioned last time what gene it was cloned to. If you remember, there was a light harvesting antenna. There was the one that was a lot of it. The one that was the inner one that we knocked out the outer one, which was 10% of the physical weight of the organism. Well, it'll probably make a lot of your things. Logical, that's working. The real question here is how can we get at this number? Because in an engineering world, it's your job to translate stuff to numbers. How can I figure out how fast water is going through a membrane? Simple concept here is that if I take a membrane like this, a vesicle, this is now an artificial cell, if you want to think of it there, 5-lipid layer, and I put the membranes in there, and then all I do is I actually measure the size of that. As water leaves it, it's going to get smaller. As water goes into it, it's going to get bigger. I just need now a means by which to measure the size of little vesicles. How do you do that? Anyway. Microscopes, it would be painful. Okay, but I want to do it in real time, and I want to do it really fast, because remember, there's 10 different water molecules per second going through there. What you have to do is you have to expose it to a change in osmotic stress. That's done with something called stop flow. Take 2 syringes, and you kind of run them beside each other at different rates, and so you have a steady state created of the residence time until those came in contact with each other with a change in the osmotic pressure. So now I just created a flow system that gave me that. And then I use dynamic light scattering. Dynamic light scattering is a way to get sizes of particles on this type of scale very accurately, in large numbers, rapidly, and so forth. Only problem here is that now I need to know how many of them are in there, right? Because all that does is tell me that it's going fast, and then I have to guess, or roughly, crudely calculate, how many aquaporins are in there. And that was essentially the hard part. I'll throw this out. I think it's kind of cool, and I like it in particular because it was the student's idea, not mine. Okay, I didn't have to do this. The device, now here's a stop flow. That, I'm not have to do that. Just run two different, the proteolipizome is a little vesicle tubing here. They go into a mixer, and if you think about it, as I drive those together, at the point of where the light is going through, it always has exactly the same time. It's like a fixed, even though it's a continuous flow system, it's a fixed point in time as a result of the residence time of flowing through there. Reactor Engineering, in case I go over that. Okay. With that in mind, okay, now I got to figure out, okay, so you get some things, you fit it to some curves and so on and so forth. Another work I want to, yeah, you kind of imagine that how fast it shrinks tells you how much volume is in there. And then you back out from the volume how much water is inside a sphere. And from that, now I know how fast it's transported. Here's the tricky part. This is a transfocal microscope in a methodology called fluorescence correlation spectroscopy. And what it is you have the laser beam kind of going in here, and it's going through, and it's literally focusing on a 6 femtoliter volume. Okay, femto, small. And within that volume, what I am doing is I am hitting it with, you guys kind of remember how fluorescence works. Do you want to tell me how fluorescence works roughly? Yeah? Boom, goes off, right? Okay, so what I can do here is that as said molecules making its way through here, okay, I can hit it with excitation. And then the question is, okay, I built into this light path a time lag of the hit it and look at it. Okay, hit it, look at it. If I hit it and it's a fast moving little critter, okay, it'll be out of the flow path before I see the fluorescence. Okay, and so you can kind of think of that as this is the fast one. Okay, and if I'm me, okay, bigger, fatter molecule, okay, and I get into here and I'm going kind of slow, I will get both excited and fall asleep all before I get out of the 6 liter volume. Okay, so I literally now have a mechanism by which if I have a tagged molecule, like green fluorescent protein, those things that you, who here has never heard of green fluorescent protein? Admit it, please. Oh, okay, so it's that. I see your hand going on. Yeah. Okay, so you're saying I never heard of it, right? Okay, so it is a famous thing of a jellyfish. Okay, and the jellyfish has that kind of iridescent cool glow. Okay, that's because it has that, and in particular, it has a protein that makes a nifty little barrel structure. That when the light molecule comes in of the right wavelength, it excites it, and then it releases it off. And so green fluorescent protein, people love to do it because you can take that little thing and you can tag it onto anything and make them glow. So if you can think of this, that if I tag aquaporin, they will have little green beacons on them if I hit them with fluorescence light. Yeah?
How big is the actual aquaporin structure? Do you remember the fact that ChE, like such a secret protein that kind of recruits the actual ChE?
Yeah, it's big in the sense that it's about 800 base pairs divided by 3 for amino acids, and you guys can do the math there. So I can't forget. It's 90 amino acids-ish. Okay, 80. Okay, which is still pretty big, right? Okay, and so now aquaporin is, by the way, if you saw the little molecules that I blew by, really big. Okay, remember it's a membrane, it's a membrane protein, and not only is it a membrane protein that has hydrophobic, hydrophilic, hydrophobic, right? That's what you need to be a membrane protein. Water, liquid, water. Okay, but it also has to have a structure big enough that it actually is a tetrameric form. So these are 4 pores, and so it actually goes into that, and then that's what sits into the actual membrane. So we're talking about now a pretty darn big molecule with a little tag off of here that would represent the green fluorescent protein. So yeah, an inherent assumption of all the work that you do with a tag of any type is that you hope you don't part up the function with the tag. But you're smart about it because you know where this is in the molecule. And you know all the rest of the others, so when you add your tag in, you add it somewhere that it doesn't mess with the function. Okay. Plus, you can do it with the same rough amount of material, stick it into the dynamic light scattering thing, and make sure that it doesn't screw it up that way. So now all I need to do is deal with this kind of fluorescence here, where I do the work with the slow guy, and then all I do is I put a cleavage site in so that I can actually now cleave off my fluorescent protein, and then I can measure how many individuals they were. Okay, and so now I can essentially get a measure of how many big ones there were, how many individuals there are, and then ultimately give a number, the number of actual pores that was in there. Because when you do the tagging and so forth, you have some information about that. There's always a relative term in there that you can then normalize out that information. So now I know the number of pores. I know how fast it was moving water in or out, and then I just divide through and it gives me the number of how many per pore. Yeah, so it's a lot of aspects to this. You know, you look at it and I go, well, that's pretty easy. Okay. True. The calculation in the end is basically algebra. Okay, it just happens to take 3 or 4 years to do all the rest of the background work that gets worked out. Okay, so there's the functionality and so forth. Oh, there's a little contour for volume. And yeah, a lot of mathematics of correlation, spectroscopy, how it works to tell you. It's measuring a diffusion coefficient. I think it's the easiest way to think about it. It's measuring how fast a molecule is dancing through 6 liter microliter centiliter volume. Okay, so I'm not going to talk about any of that kind of stuff. Or that stuff. These are some other kind of interesting things. This production, remember, we talked about this because it was a production platform. Making aquaporin was not really the goal. It just happened to be a good membrane protein to study. Okay? There are, if you want to think about it, the membrane proteins are kind of like the important ones. There's an awful lot of enzymes in there, Pac-Man chewing on this and that. But the porins are the ones that move stuff in and out of cells. Okay? And so as a result, this one here happens to be cellulose synthase, for example. You know, the cell walls are outside of the plant, okay? Cytoplasm. And so there has to be a way in which it's actually doing the synthesis of the strands to make the cell walls on the outside, you know, from the inside, if you will. And a lot of people are interested in cellulose synthase. Why? It's they're really interested in the whole process of cellulose. Paying a big center on campus, between $20 million a year on studying cellulose biosynthesis expression in the biology context. So we helped them actually do some things proving that it worked. In this particular one, it had a cofactor of diGMP, and a lot of times if you want to prove that you've actually done something, there's nothing quite like biological function. It doesn't work without cyclic diGMP. You throw it in there, boom, function happens. Well, that doesn't just mysteriously happen without there being the actual functional thing. Remember, the hard part here isn't even necessarily, you can throw all these genes into E. coli. And E. coli would make them. This would be all dysfunctional. It wouldn't work. When it doesn't work, it goes from being a functional protein to being dog food. Okay. It isn't worth anything more than any other protein that we might find. This is interesting. I just throw it out there just because you guys like write it. Believe it, and this explains things that always boggled my mind, like tuberculosis. How do people get tuberculosis for their entire life? You go on a plane trip, unfortunately sit behind some person, some subset of the population. If you get TB, you will have it and you will never get rid of it. Okay. How does that work? There's these things called persister cells. So you can basically whack it with antibiotics. You can kill 99.99% of the thing, and in the bacterial world, remembering that one E. coli can grow to the weight of the Earth in 2 days. Okay, do the calculations. It actually works out. Okay, having one left is like having a billion left. Okay, when it can come back, right? Bacteria have figured out a very scary birth control strategy. And that is when life gets really, really rough on us. Let's say we do something stupid like, I don't know, crash the Earth. And the resources all get super scarce. What is the simple solution to the problem? Genocide. Right. I mean, you just kill 99.9% of the population, and most of the problems will go away. The problem is choosing that 1% that's the same. And what crazy is? My kids made me watch. Yeah, right. Yeah. So isn't that a wild ass ending? You kind of go like, oh. Yeah. Okay. And then you're sitting there thinking, who was the bad guy in this movie? I'm not entirely sure. So this is literally a stochastic genocide gene. Bacteria do this all the time, it turns out. They make a toxin to kill themselves and an antitoxin. Okay? So they are constantly making two genes that, if mixed together, will kill themselves. Under stress, so the toxin's always there, This was the one preventing death. Okay, I mean, this is kind of like, think about this. It's kind of like, been a Russian roulette here. Like I cocked the gun and then kind of running around. I'm stressed. Ooh, I wasn't the one that was loaded. That's what they do with these things. And under stress, the antitoxin tends to go down, meaning most of the cells kill themselves. A small subset of the population then actually doesn't pull down that thing, a very small part, and they get to live. So under situations where I have, something like a TB, I give them the antibiotics for the TB. I kill 99.9% because the stress associated with that. The other ones kind of hunker down. It's hard to kill somebody that's not from me. Don't notice me. Okay. And that's the system. So this was actually very hard genes, by the way, to study. Because when you start to put these in E. coli, what do you think happens? It kills them, right? Okay. How does it work? It punches holes in membranes. The thought was, well, this organism's got shitloads of membranes. What a better place to try to express one that you can't express in a normal organism because there's so many off targets. And so this was a thing where we actually went through and actually expressed it in Rhodobacter. And you can see it gets kind of funky and upset about it, but it actually really did make the protein that we couldn't otherwise make in a bacterial system because it's a toxic gene. It's an inherent problem if you want to study a toxic gene in a bacteria. You have to actually figure out a way to actually express it. So at any rate, remember, this was, again, Rhodobacter as a production platform. We chose it for membrane proteins. Why? Did we choose it for membrane proteins? Just take a shot of articulating it. Here. Okay. Why did we choose this platform? let's say that you were going to get a quiz, and here for the benefit of you having come for Friday afternoon, on a, oh shit, Monday I have jury duty. Okay. You guys will know not to come, okay? But anyone who didn't come here, I'm not going to tell them and remind them that I have jury duty on Monday, and so I will not be here. On Wednesday, wait, I could get a quiz. How about this? If we beat Ohio State, we don't have a quiz on Monday. Okay, if we lose to Ohio State, there will be one. You'll be depressed anyway. Okay, so if you're already depressed, I'm going to give you one, and you'll at least know it's coming, so you'll be less depressed. Yeah, okay. But let's go one step further, and I ask the question, remember, this is all about platforms. You're choosing an organism. You ought to know why I've been throwing at you options, right? They're options of what they do, okay? Why did we choose Rhodobacter? To do membrane protein expression. We just went through 4 or 5 different proteins here. The class of protein is what? Great. What is the class of protein that we decided Rhodobacter was a good candidate? Yes, I do. Okay, 10. Okay. We chose, we said, here is a membrane protein. We don't express it in E. coli, because E. coli will be like, just turn it into a little gob and shove it into a little ball in the middle. It's called an inclusion body. So when E. coli overexpresses something that it doesn't quite know what to do with, it just kind of like throws it into it, balls it up like a wad of paper, and it runs it into the side of the cell. So it's called an inclusion body, which now you got to figure out how to unfold the thing, stick it into a membrane, and hope that it's going to actually work again. Why?
Rhodobacter naturally had a lot of initial tetrameric proteins.
And you know why? Under what conditions? Let's go from the 6 to the 10. What was the magic about this that made it? Yes, so you needed to induce that condition. So when you induce it, you can grow it with sugar, you get it up and rolling, have your cell going. When you do a transition from there to anaerobic photosynthesis, it'll all of a sudden start to make it on a membrane, because it needs to. And so under that induced condition now, I have a means by which to have them make the membrane protein when there's a lot of membranes around to put them into to keep them functional. That's the magic of it as a platform. Now, just for the sake, I'm going to go here. Platform. Oh, hey, look at that. This is a platform product decision tree. Yes.
Real quick, so it doesn't always have 10% of its weight in that protein. It's just under those conditions.
It's right. Oh yeah. No. Yes. So under photos, if you're feeding it carbohydrates and it's growing like heterotropically, it will make any. Well, why would it? It's all under regulation, and that's why we use that promoter. Yeah.
Why wouldn't aerobic photosynthesis?
The aerobic photosynthesis, well, first of all, it's questionable whether it's even capable of it. This organism does not really do photosynthesis. Okay. So organisms that are anaerobic photoheterotrophs. That's a lot of mouthful. An anaerobic photoheterotroph is incapable of producing oxygen by water splitting, because it only has the first step in the Z scheme. Remember I kind of told you in the algae world, is that Z scheme, you hit it with one thing, one photon, and it bounce it up. Not quite enough to split water, it trickles back down, next photoreaction center, boom, hit it again, apart, ooh, I got enough juice here to split water. That is the Z scheme associated with oxygenic photosynthesis. These guys only have the first half of the Z scheme, which has an upside down D. Okay, so it doesn't have a name. It's called anaerobic photosynthesis. Not too surprisingly, this is what I'm getting at here. So, platform product decision tree is a lot about the process. What are you? If I'm an academic, I kind of like to do what I know how to do. If I'm good at E. coli, I'll do E. coli. If I'm good at algae, I'll do algae. If I'm good at fungus, I'll do fungus. And I'll hope I can find what hammer I can slam those onto. And you'll look, so that's this. Now, over here, you start with a product. If you want to make money, okay? And then you're saying, which one should I pick? Okay? And that choice of picking keeps getting larger every day. Okay? And so, as we sequence these things, it only costs these days I just priced this out at the sequencing center here. I can sequence 4 microorganisms for $1,400. That's it. That's not too surprisingly, then, and remember, there are machines. If you ever go up to Boston, find your way to the Broad Institute. Stick it in your little walkie thing, whatever. And you go, they have a big, giant foyer slash, thing. We walk in there, and literally it has these screens, which I don't know how to do it, but they're wavy, and they're like 20 feet long and 12 feet high, that are all the DNA sequencers behind in there that are running 24/7. And you can just see this fragment going into there, and as it goes through, you see it, ATGs and like, and then you start to see it goes into ends, which means that it didn't quite have a definitive thing there. Then that one stops, then a new piece of DNA hits the machine, and you've got just these things running as fast as you can, all the machines going there, and you're like going, what is going on here? And then there's little explanations all around. And when I was there the last time, they were doing elephants. It's like they had, these things were cranking away. The Asian elephant was going here, and the African elephant was going there, and the mastodon. And I'm like, this is really cool. I mean, and they actually have an iPad like the size of this desk here that you can ask questions. So if you're a real geek and you're in Boston, definitely go to the Broad Institute. Because every one of those organisms, as they come out, an elephant may not be the best place to produce proteins, okay, unless they're big turd proteins. But that is really the decision process that I was hoping to instill in here. The thought process of thinking beyond yeast, shell, E. coli. They are the 90+% of the production platforms that are out there. The next generation is, it's all about trying to figure out how it's cheaper, certain advantages. Wouldn't it be nice if you didn't have to, I don't know, buy sugar? Okay. And you could use CO2. That may have an economic advantage at some point. So that is the big picture sort of take home on this. The last one that I wanted to hit here is less because we've seen this product before, but I did want to re-emphasize. Where did this come from? Votococcus, yeah, Votococcus browni grace B. It happens that project. It's an algae. It's an algae that makes a prehistoric hydrocarbon. So it was way back in the algae when we were talking about making it. And we also had it in the cellulosic biofuels. It was genetically engineered to the yeast, so the yeast would make it as well. And here is, what about Rhodobacter as that? Now, the choice of Rhodobacter, I will fully admit, was partially because I was working on it already for membrane proteins. So I'm an academic, right? I'm kind of a hammer trying to figure out where to swing it. The neat thing about it is this is a hammer. This is a multi-tool. Okay, it's got little clippers and hammers and knives and everything else they steal for you at the airport. And this particular project that was out there, they wanted to figure out how to use low voltage photovoltaics to make hydrocarbons, okay, biologically. And it turns out that this organism is perfectly happy to eat hydrogen and oxygen. All you need is enough photovoltaic energy to split water, which isn't that much relative to a power plant. We're not talking about 120 volts is necessary to actually split water. Okay. It's a couple, 1.2 electron volts or something. I don't know. You guys know Wikipedia. It sounds about right. That's not much. Okay, so if I can just split the water, and I get hydrogen and oxygen, I mix in CO2, it's called chemolithoautotropic growth. So it's a chemolithotrope. Okay. Another one that won't be in your biochemistry book. It's an organism that literally it runs photosynthesis with hydrogen. Right, I mean, all you got out of the hydrogen, out of photosynthesis, was to shake loose some electrons, right? What better source than hydrogen to be giving you electrons? So as long as I just have a hydrogenase that can actually use that H2, dump those electrons into the thermostat guide for quick carbon fixation. Calvin cycle, Calvin-Benson cycle, then Calvin-Benson-Grasham cycle. So it's now, I don't know, someone went back and figured that maybe the person that actually really did the work didn't have his name on the cycle. And then they probably went back one more, and there was probably a woman on it that really did the work. Okay, so they had to add her name too, so it's Cal CBB cycle now. Okay,
Or
just
hyphenated.
so it's not the Calvin cycle. It's now the Calvin-Grasham-Benson something cycle. But it is basically capturing CO2, and the process of doing that is highly inefficient. Remember back to you how many molecules per second are going through the glucose hydrolysis? Molecules per second. Right, good. Okay. The one responsible for carbon dioxide fixation, anyone know its name? The most prevalent enzyme on planet Earth. It sounds like cereal. Rubisco. Okay. Anyone ever heard of Rubisco? Yes, no? What about your wiki list for the lecture today? I mean, literally, if I was a Martian coming in here and I'd look and say, what's going on Earth? And they'd be like, oh, Rubisco, there's shitloads of it everywhere. Okay? Because it's a crappy enzyme. Okay? If you have a crappy enzyme, you need more catalysts. And its turnover number is about 4 on a good day. Worse yet, every time it does 4, one of them it screws up. Okay? And not only that, the energy it wastes burns part of what it did and the good part of the other one. I mean, this is like the real turd on your senior design project. Okay? It's actually not only not doing anything, but it's actually doing bad things for you. Okay? So, Rubisco is in that cycle. That's the magic. You've got to get electrons onto that one so they can reduce the carbon dioxide and make there's the process. Let's throw it out there. And photovoltaics, we kind of understand this a little bit. You may have done this little high school project where you took a little tiny solar cell, gave you a minuscule amount of platinum. Platinum activation or anything. We don't need much voltage because it doesn't really split water unless you not only have the voltage necessary for it, but have the over voltage necessary to actually get into the activation energy. Platinum is exactly allowed to do that. Okay, so at any rate, the electrons come from here. You can actually then do the splitting of the water. Well, there's the other side of the equation, the oxygen, CO2, and then ultimately genetically engineer the hydrocarbon biosynthesis into the molecule, which we talked about before. So here you go, literally, from sunlight to hydrocarbons in an organism capable of eating hydrogen to do photosynthesis and fix carbon dioxide. Again, I'm trying to emphasize the cool aspect of nature is it has figured out a lot of cool things that I can do this inequality. I bring up this again because I keep talking about it. You've got to get separation. In the fuels industry or in that, you've got to facilitate cheap separation. If you can't facilitate cheap separation, if I've got to boil off my thing, okay, you're already starting a big hole as far as economics and process and so forth go. And so phase separation gives you something that's nice. Gives you an idea, then the Prokaryote hydrocarbon here is at 43 kilojoules per gram relative to ethanol. So obviously a lot higher density as well. I don't want to go through all of this other kind of stuff here. Lots of nifty experiments and so forth. And I wanted to get to the concept here before you escape. And it gives you a little idea of the complexity of the rig that we set up here. Because we are literally feeding a potentially explosive gas mixture into this. Okay? And so you need to be very careful about that. And the problem is your functional constraint is that there's not necessarily an outlet gas at all. This is a fermenter with a 100% gas conversion, potentially, if it's run properly, right? There's no reason it can't use all the carbon dioxide, the oxygen, and the hydrogen. So there's no outlet gas. Okay, it's called a dead-end fermentation. There's only been about a dozen of these ever done. The biggest one was ever done was in Japan, about 300 liters. In part because people are like, it's an explosive gas mixture. Okay. Now, there's ways to get around explosive gas mixtures if you're a chemical engineer. How's that? Because if I don't give it oxygen, the process doesn't work. This is an aerobic hydrogen utilization. That's what actually gives it the efficiency of that. I can find organisms that would do the hydrogen utilization anaerobically, but without the oxygen present, they're going to have to steal that oxygen from some carbon source or something else, and it's a really inefficient process. The explosion limits, right? Things don't just explode because they're together. Plus, if I have water in there too, water vapor, that also dramatically depresses the explosion limit. You just have to know your operating region of, oh, it'll explode here, it won't explode there, and control it, but with the constraint of stoichiometry and Henry's law, because each one of these has a different mass transfer coefficient associated with the driving force. So you got to basically have the partial pressure at the right driving force, okay, for the stoichiometric delivery of the gases and everything. And it turns out it works. It actually is possible to be in the non-explosive region, operating at the right mass transfer coefficient, at the right partial pressure, so that's stoichiometric delivery for oxygen transfer. This is like engineering on steroids. Okay, it sounds like it's all biology, but the real solution is really all engineering. Because otherwise, if you were just a biologist, you could just blow a shitload of gas through there. Just blow it all through there, let it go up to the fume hood, and never work anything bigger than would blow up. Okay, like it did. Be like, oh, I did it. I made 1 femtogram. And so essentially, this is the concept of engineering here. But I wanted to go to this. If you're going to choose a platform, it's kind of like choosing a significant other. Okay. You've got to decide what maintenance coefficient you are willing to tolerate. Okay? Okay? Because if there's a mismatch in the maintenance coefficient, you're in for a long life, okay? Or a short relationship. Take your pick. The premise of this particular project is that as a biologist, I will always choose the one that grows the fastest. It's easier to do the work, and grow it up, and it just feels right? If I wanted to choose a chemolithotrope, okay, there are many of them. The one that everyone likes is Ralstonia eutropa. It is the model that they first did the genome sequence, and everybody works with it. Not too surprising, it is the fastest growing one, so on and so forth. And the premise that we threw into the monkey wrench here for this program was a fast-growing organism is likely to have a high maintenance coefficient. And we are constrained in the operating condition space as an engineer to be working where it allows us to work. Okay? It's kind of like the situation with algae under high density, right? You know, we didn't care that they were super fast growing because they were so high density they were light limited. Under these circumstances, not too surprisingly, I'm going to wake you up there. Okay, you have to throw it back in there. I can't find a piece of chalk. Okay, here. Okay. Okay. Where was that? The light limit. Okay, we're in the light limited situation there. We really were worried about what parameter? Yield, right? It was all about what their yield was to energy and so forth. I looked at it this way, and Rhodobacter, it turns out, is maybe not the world's, it's a lot slower growing organism. The fundamental question is if it is slow, it may have a low maintenance coefficient. This process is going to be mass transport limited based on partial pressures and Henry's law and so forth. I can't just simply grow this thing flat out. I'm going to have it at high density, and that high density and the density I can achieve will be dictated by mass transfer. Yeah. Okay. And so he asked so, what is a maintenance coefficient? Good question. Okay. Okay. How many girlfriends did you have? Okay. That's one way. It's a sample. Okay. So, if I take a look at the energy that's being sapped out of me, or in this case, the sugar that I'm providing, okay, this is growth, right? Okay, so this is the, if you want to look at that, this is the sugar consumed by growth. Okay, and I know that because this is the amount of growth, and then a yield is a cells produced per sugar, 1 over that sugar consumed for that. So I'm just getting it in the right units. Okay. This is kind of like the total sugar, and I'm going to put sugar in quotes because we didn't have to sugar used. And so if there's more being, if there is a sugar being consumed that is not for growth, it's just maintenance. Okay, and so we'll call it M. Okay, and so this is the maintenance coefficient. Now, the question is, which one is important? If I'm a really, really fast-growing organism, then my maintenance kind of goes away. Okay, because it's a little tiny amount of my energy is to keep, you know, this beautiful facade in front of you. Okay, okay, no, I shave once a week. Okay, so my maintenance is kind of low. If I'm a slow-growing organism, that same maintenance coefficient now means that I'm spending most of my time in maintenance and not actually doing anything productive. What I'm saying here is that this becomes process constrained. And so if this becomes process constrained, then I really do care about this, because keep in mind, in this particular case, I am feeding this hydrogen, a very expensive molecule. So all of the energy that goes to maintenance is not doing anything useful for me. It's just keeping the bugs alive. And so I need to actually go through and make a plot of this thing where I actually plot Q versus mu, which all of you know that when you have a chemostat and you run it at a constant flow rate, okay, it goes to certain circumstances, the growth rate is equal to the dilution rate. Yes, no, maybe. Yeah, this is a chemostat. Okay. It's a simple mass balance, chemostat. Not too surprisingly, that if I'm diluting at a certain rate, dilution rate, by the way, is equal to F over B. Okay. find this. I just want to, if you guys haven't had this, you should have. Okay. You've heard chemostat, right? Okay. So if you've heard chemostat, you literally can force a certain growth rate just by the flow rate. So that gives me the points that are here. And so what I do is I grow it at slower and slower rates and so forth, and I extrapolate to when there's no growth. And if I'm extrapolating to the no growth, then it's a maintenance coefficient. Okay. So that's the concept of the maintenance coefficient. Where is it?
Do you want the lower? I'm thinking you want the lower or higher?
You want it to be low. Okay, this is burning money. Okay, so you want it to be actual low. The moral of this story, now first of all, with sugar this is easy. With an explosive gas mixture, this is not quite so easy. And all of that was actually set up, and we actually did the measurements and so forth. We did it for both of the organisms, and not too surprising, the fast-growing one everyone else was using had a very high maintenance coefficient. So high, it was burning most of its hydrogen and its energy to just simply stay alive, not actually make product. In our case, our slow-growing one is actually way lower maintenance coefficient, and as a platform, wasting a fraction of the energy based on its maintenance utilization. And so the choice of the organism, like if that's why you saw the tortoise and the hare, who wins the race? The tortoise. Okay. Okay. So now you have, so the tortoise wins here too. The slower growing organism actually has a kinetic yield advantage based on this case, not on yield, but on maintenance coefficient in terms of energy utilization. That is. Okay. Yeah, you win. Hey, you too.